and there's a moment of science, or of silence, I guess. All right, so it's my great pleasure to introduce to you today Wembo. Wembo's going to be defending today his thesis, and he's going to be talking about his methodology that he's been doing the past, 4 or 5 years in our lab. Wembo is the second person that's graduating this year, Gleb being the first one, and the third person that's graduating from our lab the past couple of years, with Rob being the first one over there. Okay, so I'm gonna keep this very, very short because Wembo's got kind of stressed, as you guys can see, and I'm gonna give you more feedback about Wembo afterwards. But the plan is Wembo's gonna do the first. He's gonna present, and it's gonna be the first part of the public for everyone to ask questions. Then we're gonna invite everyone but the committee to sit, and we're gonna ask. The committee is gonna ask Wembo questions, and then we're gonna invite Wembo out, and then the committee is gonna discuss, and then hopefully, things will get done within a reasonable amount of time. Follow the procedures. So without further ado, Wembo.
All right, thanks. All right, thanks for the introduction, and thanks everyone for coming here today. So my name is Wembo Shi. I've been working with Professor Bogdan Pasaniuk in the past few years, mainly in the field of statistical genetics. In particular, I'm developing computational methods to analyze large-scale genetic data to understand the genetic architecture of complex traits. And today, I'll be talking about some of the methods that I have developed over the past several years. Okay, since this is a public defense, I would like to start by introducing a few of the basic concepts in genetics. The first concept that I'd like to talk about is single nucleotide polymorphism. So what's a single nucleotide polymorphism? So if you collect a number of samples from population, you go ahead and measure their DNA sequence. What you're going to find is that about 99.9% of their DNA sequence are similar across two individuals. However, at some position on the genome, you're going to see a single letter variation. For example, at this third position, we see that the person in the middle has a C allele, whereas the two other persons have a T allele. And we call that there's a single nucleotide polymorphism, or SNP, at this position. So a related concept to SNP is linkage disequilibrium. And in layman's term, it refers to the correlation between the SNPs. So we say a SNP is in LD if two SNPs are correlated with each other. And if you compute the correlation of some SNPs across the population, you're going to see this heat map. So here, each cell represents the correlation between two SNPs. You see this block structure, and that's because SNPs that are close together are usually highly correlated, and SNPs that are far apart from each other are less correlated. So although SNPs only account for about 0.1% of the entire human genome, it's the 0.1% of the variation plus the environmental variations that give rise to the phenotypic variations that we see on a day-to-day basis. So then why are we interested in studying genetics? Well, genetics is really the fundamental unit of the human body. And by understanding the genetic contribution on complex traits, we'll be able to understand the biological mechanisms behind complex traits and diseases. And better understanding of the mechanism can definitely lead to better diagnosis as well as better treatment of the diseases. So a very powerful, conceptually very simple, but yet very powerful approach to study the genetic contribution of complex traits is genome-wide association studies. So how does GWAS work? So in a typical GWAS, you'll first collect some samples from population. You go ahead and measure their phenotypes. Then you go ahead and measure their DNA sequences using next-generation sequencing or genotyping arrays. After that, you do an association scan. You associate each SNP with the phenotype of interest. And by association, you simply plot the phenotype on the y-axis and the genotype on the x-axis. You fit these lines through the data points, and the slope is what we call the GWAS effect size. It's marginal effect size of the SNP on the trait. Then you go ahead and do it for the second SNP, do it for the third SNP, and so on and so forth. And because both the genotype and the phenotype are measured with error, you need to account for these uncertainties. And we usually use p-value to assess the strength of association. And if you plot the p-values across all the SNPs, you'll get this Manhattan plot, where the x-axis represents the position on the genome, and the y-axis represents the negative log p-value, which is a measure of the strength of association. And this dotted line represents the GWAS significance threshold. Usually it's 5 times 10 to the negative 8. And if the SNP has a p-value less than that threshold, then we call that the SNP is significantly associated with the trait of interest. So over the past decades or so, thousands of GWAS have been performed. What's really nice about these GWAS studies is that the GWAS consortia that study these GWAS publish their summary stats. And by summary stats, I'm referring to the effect size of SNPs on the trait. So essentially, it's the slope of this line here. So the table to the right is a table of GWAS summary stats that we have accumulated over the past several years. And as you can see, most of these GWAS studies are well powered, with sample size ranging from tens of thousands to nearly hundreds of thousands. Then this begs the question, what can we learn from this set of GWAS summary stats that we have compiled? So over the past several years, not decades, I have developed several methods to analyze a single trait. I have also developed methods to analyze 2 traits jointly. And recently, we've been working on a method to contrast the genetic architecture of 2 populations together. At the beginning of my PhD, I've also briefly worked on a multivariate Bernoulli model for haplotypes. So although I have worked on a variety of projects over the past several years, they all focus on the central problem of modeling and accounting for linkage disequilibrium in analyzing GWAS studies. And apart from developing methods to analyze GWAS data, I've also applied these methods on 30+ large-scale GWAS summary stats datasets. These traits we have analyzed range from anthropometric traits, autoimmune diseases, psychiatric disorders, blood phenotypes, metabolic phenotypes, as well as education attainment phenotypes. So because of time constraints, I won't be able to talk about every project in detail. So in this talk, I would like to focus mainly on the 3 main projects that I have worked on during my PhD. So in the first part of this talk, I will present methods to partition heritability by genomic regions while accounting for linkage disequilibrium. In the second part of this talk, I will present a method to partition genetic correlation by genomic regions, also accounting for linkage disequilibrium. And third part of this talk, I will present our latest work to contrast genetic architecture of complex traits across 2 continental populations. So first, I will talk about our work to partition heritability by genomic regions. So first of all, what is heritability? So in genetics, we often model a complex trait as a sum of its genetic component and its environmental components. Then we define heritability as the proportion of the variance of phenotype that is due to genetic variations, right? And if we further assume that the genetic components and the environmental components are independent from each other, we can decompose the variance of phenotype as a sum of the variance of the genetic components and the variance of the environmental components. So just to better understand the concept of heritability, let's look at a concrete example. So let's say this blue bar represents the variance of height. Then through twin studies, we're going to find that 80% of the variance of height is due to genetic variations, and the rest, 20% of the variance of height, is due to environmental variations. Now, when we look at the SNPs found through GWAS of height, so this is a SNP that passes the GWAS significance threshold, what we are going to find is that this set of SNPs only accounts for about 16% of the total variance of height. That leaves the rest, 64% of the heritability of height, unexplained. And that's what we call the missing heritability problem. So where does the rest of the heritability come from? Well, as we start to relax the GWAS significance threshold, we're going to include more and more SNPs in our model to estimate heritability. And if we completely drop the GWAS significance threshold and use all the SNPs to estimate heritability regardless of their significance threshold, what we're going to find is that all the set of SNPs explain 60% of the variance of height, which is much closer to the total heritability of height. The reason is that although the SNPs fails to pass the GWAS significance threshold, they still have an effect they still have a small effect on height. However, we cannot detect their signal because of limits of power. And we call this type of heritability the SNP heritability. And just to recap, SNP heritability aggregates the effects of all the SNPs regardless of their statistical significance. So here, what we are interested in doing is to partition this genome-wide SNP heritability into each region on the genome. And you want to figure out which part of the genome contribute a disproportionate amount of variance to the traits. So let's say this is our, so what do we mean by partitioning heritability by genomic regions? So let's say this simple diagram represents our entire genome. And on this genome, we have different kinds of SNPs. So we have SNPs in the coding region. We have SNPs in the DNAS1 hypersensitivity sites. We also have SNPs in the transcription factor binding site. So these are what we call the functional SNPs, which usually have higher effect on the trait than the rest of SNPs. So what do we mean by partitioning? So we partition the genome into multiple regions. And instead of asking what's the variance of trait explained by genetic variations at all the SNPs, we are interested in asking what's the variance of trait that is coming from genetic variations at each of these small regions on the genome. And we call it the local SNP heritability. So as you can see, at the small region on the genome, few SNPs are likely to be causal. Few SNPs are likely going to have a large effect on traits, and most of SNPs are likely going to have a small or zero effect on traits. So a lot of the traditional methods that estimate heritability assumes that the effect of every SNP is drawn from the same distribution, or in other words, they assume that the effect of SNPs are on the same magnitude. So although this assumption is very robust on genome-wide scale, at the local level, it's not very robust. So in order to solve this problem, we resort to the fixed effect model. So how does fixed effect model work here? So let's say this equation represents our phenotype, where X is the genotype vector, beta is the effect size vector of SNPs on trait, and epsilon is the environmental effect. So we say the model is fixed effect model because we assume that beta is a fixed vector rather than a random variable. And by making the assumption, we don't explicitly say that every SNP contributes the same amount of variance to the traits. Now, under our definition of heritability, we can see that under this model, we can define heritability simply as beta transpose times V times beta, where V here is the LD matrix, is the correlation between the SNPs as represented by X. Now, if we are given the true effect size vector beta, then estimating heritability is trivial. You can just simply plug in the true effect size vector into this equation, and that will give you the exact heritability estimates. However, in reality, the beta effect size vector is not given to us, so we somehow have to infer it from data. And it turns out that we can infer this beta from genome-wide association studies. However, the lesson here is that genome-wide association study does not give you directly the true effect size vector. What it gives you is a confounded version of true effect size vector. And in fact, if you look at the GWAS effect size for single SNP, the GWAS effect size for a single SNP is going to be the sum of true effect size of the single SNP plus the effect size of SNPs that are in LD with the SNP, which is definitely not equal to the true effect size of SNP-J. And in general, it can be shown that the vector of GWAS effect size vectors follows a multivariate normal distribution with mean equals to the LD matrix times the true effect size vector plus some random noise as well. Okay, now let's recall that this is our definition of local SNP heritability. Now, if we plug in the GWAS effect size vector here, we will definitely not be able to get the correct estimates of a local SNP heritability because the GWAS effect size vector is not equal to the true effect size vector. But fortunately, we can solve this issue by simply multiplying the inverse of the LD matrix in front of the GWAS effect size vector. And that will give us an estimate that is equal to the truth in the expectation. And after correcting for bias due to finite samples, we arrive at this unbiased estimator of local SNP heritability. So we have verified this estimator through a lot of simulations, and we also apply it on the 30 complex traits that I just showed you earlier. So what does our method give you? So to illustrate the output of our method, I would like to show you the results for 4 lipid phenotypes: HDL, LDL, total cholesterol, and triglycerides. So here, the x-axis represents the position on the genome, and the y-axis represents the local SNP heritability. So each bar here represents the amount of variance in traits that is explained by genetic variations at each of these regions on the genome. So as you can see, most part of this genome is very flat. But at some regions, for example, at this region on chromosome 2, and this region on chromosome 11, and also this region on chromosome 19, we see peaks. And it turns out that these regions harbor the APOB, APOA, and APOE gene, which are known to be associated with a lot of lipid phenotypes. Okay. Now, if we zoom out into chromosomes and want to figure out what's the contribution of each chromosome on the complex trait, what do we find? So here, we're looking at the trait schizophrenia. So here, the x-axis represents the chromosome length, and the y-axis represents the heritability. So each dot here represents the amount of variance contributed by genetic variations on each chromosome. And as you can see, the amount of variance coming from each chromosome is highly proportional to the length of the chromosome. And that says that the heritability of schizophrenia is more or less evenly spread across the entire genome. And that also says that the causal variants are also evenly spread across the genome, and the effect size of these SNPs are on the same magnitude. So this is also saying that schizophrenia is a highly polygenic trait. So what does polygenic trait mean? So to better understand a trait that is polygenic, I like to contrast it with a monogenic trait. So for a trait that is monogenic, we are going to expect that only a few SNPs is going to impact that trait. An example of that is lactose intolerance traits. For a trait that is highly polygenic, we are going to expect that a lot of genes is going to contribute to this trait. And a classical example of a polygenic trait is height, which has been estimated to have about 700 causal SNPs. So now that we have a concept of polygenicity and monogenicity, how can we quantify polygenicity of complex traits? Here, I would like to show you a few examples. So let's say this green bar represents the total SNP heritability of a trait. So for a trait that is less polygenic, what you are going to expect to see is that a single region on the genome is going to concentrate all the SNP heritability of the traits. Whereas for a trait that is highly polygenic, we are going to expect to see that the total SNP heritability of the trait is evenly spread across the entire genome. Now, if we look at the region with the highest local SNP heritability for traits, for a trait that is less polygenic, we are going to see that this region is going to account for a large fraction of the total SNP heritability of the trait, say 60%. Whereas for a trait that is highly polygenic, the region with the highest local SNP heritability is only going to account for a small fraction of the total SNP heritability of the traits, say 5%. So let's keep this picture in mind. So here we computed this red bar for all the 30 complex traits that we have analyzed. So let's recall that the red bar represents the fraction of total SNP heritability coming from the region with the highest local SNP heritability. So effectively, what we have done here is that we have sorted all the traits from the least polygenic to the most polygenic. And as you can see, traits such as rheumatoid arthritis and HDL are less polygenic, whereas traits such as height and schizophrenia, they are highly polygenic, which is what we expect to see. So just to conclude the first part of my talk, in the first part of my talk, I presented a method partition SNP heritability by genomic regions, and I used these estimates to contrast the genetic architecture of complex traits. Now, in the second part of this talk, I would like to talk about a related problem of partitioning genetic correlation by genomic regions, while also accounting for linkage disequilibrium. So when you look at two phenotypes, such as height and weight, in a population, what you're going to see is that these two traits are highly correlated in a population. So people with higher height tend to also have a higher weight. Then a natural question to ask is the correlation between height and weight due to genetic variations, or is the correlation between these two traits mainly due to environmental variations? So how are we going to answer that question? Well, it turns out that if you assume a linear model for these two phenotypes, you can decompose the covariance between these two traits as a sum of the covariance of the genetic component of the two traits and the covariance of the environmental component of these two traits. And if the covariance of the genetic component is 0, then we can safely say that the correlation between the two phenotypes is mainly driven by environmental variations. However, if this part is very strong, then we can say that there is some genetic contribution to the correlation of these two phenotypes. Now, a method called cross-trait LD-square regression is able to estimate this genome-wide genetic correlation between pairs of complex traits. And if you apply cross-trait LD-square regression across 24 complex traits, you'll be able to get this heat map. So each cell in this heat map represents the genome-wide genetic correlation between two phenotypes. So the darker the color, the stronger the genetic correlation. And as example, let's look at this cell. So this cell is for the pair of traits triglyceride and LDL. So this pair of traits has a genome-wide genetic correlation of about 0.4. So what does that mean? Well, it means that if you know the genetic components of LDL, then you should be able to predict with modest degree of accuracy the genetic components of triglyceride, right? So as you can see, most pair of traits are highly correlated, and then we ask ourselves which genomic region contribute to this genome-wide genetic correlation. How can we find these regions? How can we find regions that contribute a disproportionate amount of genetic correlation to pairs of traits? So here we're going to answer this question by partitioning the genome-wide genetic correlation into each region on the genome. So here again, we have this simple illustration of our entire genome, and we have these functional SNPs on the genome. And we partition the genome into multiple independent regions. So instead of asking what's the correlation between phenotypes driven by genetic variations at the entire genome, what we are asking now is what's the genetic correlation between two phenotypes that is driven by genetic variations at each of these regions on the genome. And we call it the local genetic correlation. And as you can see here, we have the same problem where at each region, not many SNPs are going to have a large effect on traits. So we cannot use the traditional methods that assumes every SNP contributes an equal amount of variance to traits to estimate local genetic correlation. So again, we resort to the fixed effect model. So instead of dealing with one trait, we are dealing with two traits, where Y1 and Y2 are traits of interest, X is still the genotype vector, and beta and gamma are the effect size vector of SNPs on the two traits. And epsilon and delta, they are the environmental effect. Now we can decompose the covariance between Y1 and 2 as a sum of the covariance between the genetic component and the environmental component. Then on our definition of genetic covariance, we can see that the covariance is beta transpose times V times gamma, where V is still the LD matrix here. So if we are given the true effect size vector beta and gamma, then estimating local genetic covariance is straightforward. You can simply plug in these two vectors into this equation, and that will give you the exact local genetic covariance. However, in reality, these two vectors are all hidden from us, but we can infer it through genome-wide association study. And as you recall, this is our definition of local genetic covariance. And if we, as shown before, if we simply plug in the GWAS effect size vector here, we will not be able to get the correct estimate of local genetic covariance. And the reason is that GWAS effect size vector is not equal to the true effect size vector. But we can use a similar trick to account for LD confounding on the effect size vector by simply multiplying the inverse of the LD matrix in front of the GWAS effect size vector. And after correcting for bias due to finite samples, we arrive at an unbiased estimator of local genetic covariance. And we have shown through simulations that this estimator gives approximate unbiased estimates of local genetic covariance, and we apply it to the 30 traits that we have just shown earlier. So what do you get by running this method across a pair of traits? So here I'm showing you an example involving age at monarchy and height. So here the x-axis represents the position on the genome, and the y-axis represents the local genetic covariance. So each bar here represents the covariance between two traits that is driven by genetic variations at each of these regions. So, age and monarchy and height have a genome-wide genetic correlation of about 0.13. And it's not surprising that we can find many local genetic correlations that are also significant. Specifically, we find that there are, for this pair of traits, there are 27 regions that show a very strong genetic correlation at the local level. And most of them are actually all positive. So what does that mean? Well, it means that regions that increases height tend to also increase age and monarchy, and regions that decreases height tend to also decrease age at monarchy. And this could be explained by this model, where these two traits are all driven by a pathway that drives both traits in the same direction, where in turn this pathway is driven by genetics. Now, when we look at our local genetic correlation distribution, we find something very interesting. So when we look at two pairs of traits, we find that for some pair of traits, they have the same genome-wide genetic correlation. But when you look at their distribution of local genetic correlations, they show a very dissimilar pattern. So to illustrate what we saw, let's go over an example here. So here we are looking at two pairs of traits. So both pairs of traits have a genome-wide genetic correlation of about 0.05. However, for the pair of traits in the left figure, the total genetic correlation is the sum of a positive local genetic correlation plus two small but negative local genetic correlation. However, for the pair of traits in the right figure, the total genetic correlation is the sum of three small but all positive local genetic correlation. So the idea here is that although two traits may have the same genome-wide genetic correlation, their local distributions might be very different from each other. This is also true for pairs of traits where the genome-wide genetic correlation is 0. So it could be the case that all the regions have a 0 local genetic correlation. But it could also be the case that some region is positive and some other regions are negative, right? So when you look at sum, it becomes 0. But if you look at each region independently, you may still see a strong signal. And this is the case for the pair of traits, HDL and LDL. So their genome-wide genetic correlation is close to 0. However, if you zoom in at each of these regions, you will still be able to find that there are 9 regions that show strong local genetic correlation. So some of them are positive and some of them are negative. So if you look at sum, you won't be able to see the signal. But if you zoom in at each of these locus, you will be able to find a strong signal. So what does this figure mean here? It means that some regions that increases HDL tend to also decrease LDL, and some regions that decrease LDL tend to increase LDL. And possible model to explain this figure is as follows. So these two traits are likely driven by two pathways that impact two traits in different directions. And these two pathways are in turn driven by genetic variations. So just to conclude here, so in the second part of this talk, I have presented a method to partition genetic correlations by genomic regions, while also accounting for linkage disequilibrium. So up to now, all the methods that I have presented deal with GWAS data of a single population. So I'm dealing with LD in a single population. In the final part of this talk, I would like to present our very recent methods to contrast genetic architecture of two populations while accounting for linkage disequilibrium in two different continental populations. So up to now, most of these GWAS studies are based on samples of European ancestries. For example, by the year 2009, 96% of the GWAS samples are Europeans, and the rest 4% are spread across the entire world. However, this number has been changing. For example, by the year 2019, the number of non-European GWAS samples becomes 19%, and most of the 19% non-European samples are Asians. So now that we have GWAS data of both Asians and Europeans of comparable sample size, we ask ourselves, what can we learn from these two datasets? So here, what we're interested in answering is, do Asians and Europeans have the same causal variance for complex traits? So to better illustrate the concept of causal variance, I would like to go over a simple example. So let's say we have 5 SNPs here, and we use star to represent SNP that is causal, and we use a circle to represent the SNP that is not causal. So as you can see, SNP1 and SNP4 are not causal in either Asian or Europeans. And SNP2 and SNP5 are causal only in Asian or Europeans, respectively. And we also see that SNP3 is causal in both Asians and Europeans. So we call this type of SNP, so we call a SNP that is only causal in one population as a population-specific causal variance. And we say that SNP that is causal in both populations as shared causal variance. So the question we are interested in answering is, how many SNPs are population-specific, and how many SNPs are shared across these two populations? So how are we going to answer this question? Well, if you are given these causal configurations, the answer in this question is simple. You can just simply count how many SNPs are population-specific and how many SNPs are shared, and that will give you the answer. But in reality, these causal configurations are not given to us. So we have to infer them from GWAS summary statistics. So to better illustrate the intuition behind the method, let's look at a simple scenario where we have five SNPs that are not correlated with each other. And let's say we are also given this Manhattan plot for these 5 SNPs. As you can see, only SNP3 here passes the GWAS significance threshold, where the rest 4 SNPs are below the GWAS significance threshold. So from this plot, we may infer that SNP3 is causal in both populations, whereas the rest 4 SNPs are not causal in either population. However, this can be difficult if there are LD in the two populations. So what do I mean by that? So let's assume that both populations have the same genetic architecture. They have the same causal variance, and the effect size of the causal SNPs are the same across these two populations. However, if you push this effect size vector through the LD and you do a GWAS on them, what you're going to find is that the Manhattan plots are going to be very different from each other. And if you're trying to infer the causal variance based on these two Manhattan plots, you're going to get completely incorrect results. So here we have developed methods to model the generative process of GWAS summary statistics in two populations. So we start from modeling the causal configuration vector of the two populations. And on top of that, we model the effect size of the causal SNPs. And next, we model the generative process of the GWAS, which here we just model z-scores. And at the very end, we put everything together to give us a joint distribution of the GWAS summary statistics in the two populations. Now, this model allows us to do a lot of inference. And as a high-level overview, I would like to go over the input and output of this method. So the input to this method is the GWAS summary stats in two populations of a single complex trait, and also the LD matrix of the two populations. As output, we generate two outputs at different levels. So one is at the genome-wide level, and the other one is at the individual SNP level. At the genome-wide level, we output the proportion of Asian-specific causal variants on the entire genome. We also output the proportion of European-specific causal variants, as well as the proportion of shared causal variants. And at the individual SNP level, we output the posterior probability that the SNP is specific to East Asians, and the SNP is specific to Europeans, and that the SNP is specific and the SNP is shared by both populations. So we ran a lot of simulations to make sure that our method gives robust inference of population-specific and shared causal variants. And we apply it on GWAS summary stats of 9 complex traits across East Asians and Europeans. So what do we find? We find that about 60 to 80% of the causal variants in East Asians is also causal in Europeans, and vice versa. So 60% to 80% of causal variants in Europeans are also likely to be causal in East Asians. But interestingly, what we find is that about 20 to 40% of the causal variants are specific to each population, and this also depends on the complex trait as well. So just to give a concrete example, let's look at the trait LDL. So what we found is that for both East Asians and Europeans, about 1.3 to 1.5% of the genome are causal for these traits. And among this 1.3 to 1% of causal variants, 1% of them are shared by both populations. However, each population also have about 0.3 to 0.5% of population-specific causal variants. Then we ask ourselves, where are the shared causal variants and where are the population-specific causal variants? So we first looked for shared causal variants in regions that have GWAS signals in both populations. So what we did here is that we take the sum of the probability that a SNP is shared across two populations, and that will give you the expected number of causal variants that are shared across populations. So here again, we're looking at regions that have GWAS signal in both populations, and we find that in these regions, there are about two causal variants that are shared in two populations. But what we find interesting is that even if we look at regions that have GWAS signal in only one populations, we still find that there are about two shared causal variants at these regions. So it means that, so one reason that why we see this figure is that although these regions does not contain European GWAS signal, they have causal variants that are shared with Europeans. And the reason why we did not see a GWAS signal here is likely because of the LD difference between these two populations. So next, we looked for population-specific causal variants at these different regions. What we find interesting is that even at regions where there are GWAS signal in both populations, we still tend to see that some regions tend to still have a lot of causal variants that are specific to each population. And one explanation to why we see this here is that although these SNPs are specific to each population, they may be impacting the same gene, which in turn drives the complex trait of interest. And just to summarize the final part of my talk, here we have presented a method to contrast the genetic architecture of two populations, and we dissect the population-specific and shared causal variants. So before I end my talk here, I would like to make a few acknowledgements. So first I would like to thank my thesis advisor, Bogdan Pasniuk, for his for supervising me over the past few years. I'm very lucky to have Bogdan as my advisor. He always have time to guide me through all the projects, to provide guidance to my writings and presentations. Otherwise, he's really good at making my projects have a higher impact on the field. What I really like about Bogdan is that he gives me a lot of freedoms in what I want to work on, and I benefit a lot from this freedom. And I would also like to thank my committee members, Jenna Sinsheimer, Kenneth Lynch, Pavi Pavakanta, Rita Kenter, and Arash Amini. So they have taught me a lot in theoretical and applied statistics, as well as optimization methods, which have immensely helped my project. I also owe a big thank you to my lab mates, as listed here. They have helped me immensely during the course of this PhD, not only in terms of project, but also on life in general. It's really a great pleasure to have worked with them during my PhD. And my PhD wouldn't have been very smooth without the help of all these all of our collaborators, both at UCLA and at other institutions. I especially want to thank my summer students, Sarah Spenlove, Astrid Manuel, Natalie Dawn, and so on. So they have helped tremendously during summer on several projects, and I benefit from them in terms of mentoring as well. I'm very grateful to them. I also want to mention now from European Bioinformatics Institute, who have taught me a lot on depression disorders. And lastly, I would like to thank all my friends at UCLA. Just trying to look good. So without them, without their support and understanding, I won't be able to finish my PhD. I also like to thank my parents for supporting me and for their understanding during this PhD journey. And I'd like to end my talk here, and I will be happy to answer any questions that you may have.
Any questions? We have time for questions.
I have one.
Sure.
You can save it for the...
Oh, save it? Okay.
Any questions from the general?
But they're gonna wonder what the question is. Later.
Hi, question.
So, like most of your methods and derivations, so did you assume like continuous rate and like the estimator of the genetic correlation of directability, but do those formula hold for discontinuity?
So yeah, good question. So we designed our method based on the assumption that the trait is continuous. So for dichotomous trait, the method is not designed for dichotomous trait. But since we operate on the summary stats levels, we operate on the z-score level, right? So if the association statistics of the dichotomous trait is similar to that of a continuous trait, then the method is still going to apply there. So in general, it should still be very robust on dichotomous traits. But that's a good question.
So really wonderful work, and congratulations. Actually, I don't know, maybe you didn't pass.
Sorry.
I have a question for you about your local genetic correlation.
Sure.
And like how, let's say there's LD, like, local genetic correlation versus co-localization, those are like kind of different ways to view it. And your last work, you're looking at shared causal variance. So
Yes.
like, when you see local genetic correlation, do you have a sense for, do you think it's the same variance, or do you think it's different variants that are in LD? What's your intuition on this?
So that's a good question. So we have actually addressed this problem in our manuscript. So local genetic correlation does not necessarily mean colocalization. So you could have two traits that have different causal variants, but they still may have a local genetic correlation simply due to LD. So local genetic correlation is not a test for localization. It's testing for whether the genetic effect of two traits are correlated with each other. So it's different from localization.
So you think of like local genetic correlation as basically telling you if it's the same genes that are involved as opposed to like
Yes.
the same variants.
Yeah, that's a good way to view local genetic correlation.
Yeah, really nice work.
Thank you.
All right, if there are no other questions, then we'll excuse everyone.